It's my great pleasure to introduce Christa Buecker, who is currently at Max Perutz Labs in Vienna. She got her start doing her undergraduate and master's in Stuttgart before coming over to North America to do her PhD at Mass General at Harvard, where she worked on understanding different flavors of pluripotency and was one of the first to show that human embryonic stem cells, or iPS cells, could exist in different metastable states, including one more similar to mouse embryonic stem cells. She then moved on to Jono Isaka's lab at Stanford for a postdoc. And starting there, she's done some absolutely amazing work to look at how enhancers regulate their target genes and how distance impacts that, and the effect of enhancers and their regulation during the differentiation of cells or the development of an embryo. And so I'm excited to hear her new work, which I believe was very recently published, and she'll be telling us about that today. And let's give a warm, fragile nucleosome welcome to Christa. Please take it away. Oh, you're still muted.
Yes, I know. I realized that. I was thanking you. So thank you for having me. And I actually wanted to point out that as a lab, we have a very, very fond memory of the Fragile Nucleosome series, because we got our first publication ready right when the pandemic hit, and it was really the only place that Henry, my PhD student at the time, was able to present his work. And so that was one of his first starts into it. So that's why I'm like happy to be back and present another story that also involves the same PhD student who has now moved on to newer lands. But the one I want to talk about is still about enhancers, and also stay with the theme that Henry had at the time already introduced, and that is enhancer cooperativity. But I first want to take a bigger step back, and that is the main question that drives me and my lab forward. And that is the fact that we have one genome, and this genome can be utilized in many different ways to then give rise to this amazing, beautiful little boy over here. And of course, the main question that I think that drives me forward as a scientist is how these cell type specific gene expression patterns are established during development. And so, when we want to talk about gene expression, of course, we are going to start at the promoter. That's where the transcriptional machinery is established, and that's where the polymerase is taking off. And it's then producing the mRNAs that we are interested in. However, when and where this is happening, or which cells this is happening, is controlled by cis-regulatory elements or enhancers. And these elements, they can be megabases away from their promoter of interest. And in the active states, the enhancers are activated, the nucleosomes are removed, transcription factors bind, they recruit a whole bunch of co-activators that are then somehow communicating with the promoter to activate it. And so over the last years, we have really zoomed in on the enhancer. And there has been really beautiful work done over the last few years dissecting and understanding how the individual transcription factors bind to the enhancers and how they're really activating an enhancer. And for example, we and others have used massively parallel reporter assays, or MPRAs, that really could identify all different combinations and syntax and arrangements of transcription factors on DNA and how they're activating a reporter gene. These data have then really led to the de novo design of enhancers, where we now are able to really generate tissue and cell type specific enhancers that are really driving the expression in a cell type where we want it to drive. But the main issue that I see, or that I and others are working on, is that we actually cannot go back. We cannot just take a sequence and then predict yet what are the expression levels that this specific sequence would drive. And one of the main reasons for that is that when we're taking some of these reporter assays, we don't really take into consideration how the actually endogenous loci where these enhancers are located. And so when I want to cite back one of these beautiful papers by Luca Giorgetti, where they took an enhancer and then they set it free within a TAT and and generated numerous cell lines with different distances of the enhancer to the promoter, and then asked what is the expression and how can it activate the promoter. And what they found is that with increasing distance to the promoter, the enhancer loses its ability to activate the promoter. And so in other words, if we expand the space between the enhancer and the promoter, the enhancer is not able to activate anymore. However, we know in the genome that the enhancers are still able to work. And so the main focus of my lab these days is really understanding how can enhancers bridge these distances. And so the main hypothesis that we have is that on average, every gene in the human genome is not flanked just by one, or it's not regulated just by one enhancer, but it oftentimes works in concert with multiple enhancers. And so oftentimes there are up to 4, 10, even more putative enhancer elements within this vicinity. And we think that the cooperation of these enhancer elements together lead to the upregulation of the target gene, and thereby allows for really activation across a distance. And so this is not just based on some like, weird hypothesis that we have, but we have in the past, and this was the work that Henry had presented at the time, shown that at the FGF5 locus, we have numerous enhancer elements that are all working together to activate the target gene. But each of these individual enhancer elements has very low enhancer activity. So it has to really work in concert to activate the target gene. And then similarly, and many of you are maybe familiar with this work, Doug Hicks and Mira Kasuf have shown that at a super enhancer like the alpha globin locus, there are combinations of classical, really strong enhancer elements that really depend on additional weak enhancer elements to then activate their target gene. So, but these are two endogenous enhancer loci that are, still just two examples, and we don't understand how this interaction and how their elements are working with each other. And so what we wanted to address is really the question of how do enhancers cooperate with each other across distance, and what are the rules that are underlying this enhancer cooperativity. And so once Henry had submitted his first study, we took a very, very, very long time at the whiteboard to really come up with a system how we can address this question. Because just taking apart another enhancer cluster is a lot of work, and it's really tedious and takes a lot of effort. And so we did not want to go that route again and just dissect another enhancer locus. And so what we decided to do is to design a system that allows us to integrate individual and combinations of enhancers at a model locus of interest. And so Henry teamed up with a second PhD student in the lab, Songji, and we chose a mouse embryonic stem cell line that is a cross between two different but very, very closely related mouse strains. So we have enough SNPs between the two lines so that we can separate them from each other. Now we chose the beta globin locus because this locus had been previously suggested to be not active, but also not actively repressed. So we figured this could be a great area where we can have a neutral ground for integrating and testing different enhancers. And so then we chose the black 6 allele and we integrated close to the gamma globin gene, the mCherry expression cassette. This mCherry reporter can then be read out on fax as an easy readout for enhancer activity. It's controlled by a minimal promoter. And then we integrated 3 different landing pads based on the LOXP system or LOX system. at 1.5, 25, and 75 kb away from the transcription start site. We want to know how this really works and how we chose these loci and how we integrate things. Really take a look at the paper because I think it's demonstrated there really nicely how we can integrate things there. And so this locus now allows us to reconstitute enhancer clusters into a safe harbor locus, and then really rebuild from the bottom up gene regulatory landscapes. And so I was first a little bit skeptical whether this would all work, but it came together really well because we started to test the system by using the NANOC enhancer. This one is the enhancer that is closely located to the NANOC promoter. And it has all our favorite enhancer marks, P300, K27 acetyl, and a little bit of K4 monomethylation. So when we integrated this enhancer at 1.5 kb, we found that all of our individual clones showed really high reproducibility of mCherry expression. And this was also true when we moved the enhancer to either 25 or 75 kb. So all of these clones are individually derived, and they really show an absolutely amazing high reproducibility of the fluorescence. And so we can then take each of the individual clones. We can then compare them across the distances, how the NANOC enhancer might be changing its activity or ability to activate with increasing distance. And so when we do that, we can compare in these three examples how the enhancer loses some of the ability to activate with increasing distance. And the way that we're analyzing is that we're always normalizing it to 1.5 kb because we take the 1.5 kb as our intrinsic enhancer strength. So this is how we define this enhancer at this locus has this activity. And then we compare it to the 25 kb and the 75 kb landing pads and ask how does this change now over the distance. And as you can see from NANOC, it has a fairly similar activity at 25 kb, but then it drops at 75 kb when we integrate the enhancer at 75 kb. This is not surprising. This has also been shown by Luca Giorgetti. However, we can now take not just one enhancer, but we can really take a whole bunch of different enhancers and ask how are they behaving across distance. So we chose 5 different ones. Their nature doesn't really matter. And we integrated them at 1.5, at 25, and at 75 kb. And the one thing I want you to take away from this slide is that no matter which enhancer we chose, we never so far, but I actually highly doubt this will happen, found an enhancer that only activates from a distance, but that doesn't activate from close by. But it's the first thing. The second thing is they also scale somewhat with their intrinsic activity. So stronger enhancers will never really drop drastically down, and weaker enhancers will most of the time not completely lose their ability to activate. But I'm coming to that in a second, back to that in a second. Okay, so we can then compare for each of these elements how they're behaving over distance. And when we do that, we found that we have quite a few strong enhancers that are activating mCherry expression from all 3 distances, but each one of them, even the strong SOX2 SCR, and we really took the whole 6.2 kb here, loses its ability to activate at 75 kb. However, we also have a few weaker enhancers that are not able to activate so well. They have somewhat of an ability to activate from 25 kb, but already at 75 kb, they cannot activate the mCherry expression anymore. So these individual weak enhancers, they really need to be fairly close to their target promoter to then activate it. And so this then leads us to the question of what is it actually that determines the ability to activate from a distance. We think one of these abilities, or one of that is really the intrinsic strength. How many transcription factors are binding? How many cofactors are recruited? And this then scales with the distance. So if you have a very strong enhancer, that has already many goodies coming in at 1.5 kb, it will also do so at 25 kb. And so its radius to activate is good enough to reach the promoter at 1.5, to reach the promoter. However, we also don't think that's the whole explanation. Because if we, for example, take these two elements, the MAP4K3E2 enhancer and the RYBPE1 enhancer, both of them are fairly weak. And we didn't test them in the exact same experiments, but you can see that the MAP4K3 enhancer drops by almost 75%, and it's very, very lowly active at 25 kb. So much so that in the first experiments we did, it was actually not statistically significant. The RYBPE1 enhancer is slightly less active than the E2 enhancer, but it only drops by 25%. So it does not lose as much. So we think that there is differences here in the transcription factors that are recruited to these enhancers that then lead some enhancers to not drop as much in the distance. What these transcription factors are, we just have way too little enhancers right now, and we're setting up screenings right now to really go much, much wider into that, to really understand what makes these differences. At this point in time, we don't know. Okay, so this is now for the distance. But what happens if we now combine multiple enhancers? Because that's what we originally set out to set up our system for. And so this is one of my favorite examples that we've done over the last few years. These two enhancers, the MAP4K3E2 enhancer at 25 kb and the MAP4K3E1 enhancer at 75 kb, they both are not very strong enhancers. Actually, both of them at these positions are not statistically significant over the no enhancer control. However, once we then place them two in there together, now we actually do see activity. This is not a massively strong enhancer, but it's very well visible and it's highly reproducible to really activate the target gene from a distance. So this demonstrates that these weak enhancers, they can cooperate with each other to activate the target gene. These two enhancers were taken from the exact same locus, but we can also take the E2 enhancer and combine it with some of our stronger enhancers. And we've found that the E2 enhancer is able to work together with all of our even strong enhancers to lead to this super additive activation of the promoter. So we concluded that so far, all of our tested weak enhancers really show this ability to cooperate and thereby strengthen the gene expression beyond simple additivity. So they do add something to the activation at the target gene. Furthermore, we do think that this depends somewhat on the position where they are integrated. So if we, for example, switch around then a knock enhancer and place it at 25 kb and place the E2 enhancer at 75 kb, now we do not see any kind of super additive effects. However, we're also reaching a promoter plateau. So this might then not be going much more further in the activation. But for other weak enhancers, we've seen the same. So we do think that the way that the enhancers are oriented with each other does matter. So then finally, what limits now the synergistic enhancer activation? And what are parts of, like, where does it reach its limits? And so one thing that we were wondering about is we have two enhancers. Maybe each one of them brings in different things. And so when these sufficiently different co-activators or whatever it is that we really still don't understand, we might lead to this stronger activation. However, if we would bring in the same on both of these enhancers, even with slight differences, maybe this just leads to just a simple additivity. And to test this, we actually had a few cell lines available that allows us to look into that. Because we had the E1 enhancer from the MAP4K3 locus together with the NANOC enhancer, and this showed a beautiful super additivity that we've seen before, as we've seen also for the E2 enhancer. And when we combine, as I showed you earlier, when we combined the E1 enhancer with the E2 enhancer from the MAP4K3 locus, we showed a really strong super additive behavior. However, when we take 2 times the E1 enhancer and placed it at 75 and at 25, we do see a little bit of a slight increase beyond an additive behavior, but not very strong, suggesting that really it might really be that we're bringing in very similar things. And this might then lead to a simple additive behavior. So we're looking into this further and really trying to tease out what are the combinations that lead to super additive behavior and what are the ones that are just leading to simple additive behavior. And then finally, so our model suggests that enhancer cooperativity can extend the range of an enhancer. But of course, you're going to ask, well, cohesin-mediated loop extrusion and specifically CTCF have also been implicated. And so we wanted to understand how CTCF sites affect enhancer cooperation. And so this is more by chance because we actually found that in our locus, there were 2 different CTCF sites that were still in the system. We had kind of overlooked this at first, or not overlooked, we had kind of dismissed this at first because when we looked at the overall 3D genome organization surrounding the HBB locus, it showed no structure at all. So if you really have to squint to see any kind of 3D genome organization that goes beyond that would resemble any TAT here. However, we figured maybe if we introduce transcription at the locus, we might now see 3D changes in the 3D. I don't have the data for this yet. This is actually a position I'd like to fill right now. But what we decided to do is to go deeper into the CTCF sequences that were here and ask whether they have an effect. And so in order to do that, we deleted the 2 motifs one after the other. And so we started out with this peak 1, and then we deleted peak 2. Data I'm not going to show you. If we only delete peak 1, we do not see any changes in gene expression, or very, very, very little changes. But then we deleted peak 2. And now we asked, how does it actually affect enhancer activation from a distance? And so the first part here shows that if we have a weak enhancer at 75 kb, it does not show magically now activation of the promoter. And so here we don't see any differences between the cell lines that do have the CTCF sites or that do not have the CTCF sites. But if we already have an enhancer where we do have some activity, like the NANOC enhancer at 75, and we delete the CTCF sites, now we see an increase in activity at the promoter. Furthermore, if we have cell lines where we have cooperativity between the different sites, we also see an increased expression. So CTCF binding interferes with enhancer-promoter communication when we have also multiple enhancers cooperating. However, the interplay between the different regulatory sequences and the genome finally determines what the ultimate gene expression. Because if we, for example, compare here the cooperation between NANOC and MAP4K3E2, it leads to a strong synergy. But it also does so when we delete the CTCF sites. So we get an even stronger expression. So the takeaway from this is that we really have a whole gene regulatory network, or the whole gene regulatory landscape. And this plays really a crucial role in regulating transcription. We also think that the individual enhancer sequences do not alone determine the transcriptional outcome. They do work in concert with the other sequences that are there at the locus. Now, and so then as a conceptual outlook, what this could mean is that, for example, if we have an enhancer that has now to choose between 2 different promoters, if there's now such a weak element somewhere in close proximity or a facilitator, that might then actually lead to the activation of one promoter versus another one. And so we think that looking into this, into the enhancer cooperativity and into the overall regulatory landscape is an important direction that we in my lab want to take on in the future. So I'm going to take this also to do a very shameless advertisement, because we have our PhD program open right now. And so if you're interested in research in Vienna, in a beautiful city and an amazing campus, take a look at it. And I'm stopping here, and I would like to thank my lab. Henry, who was the mastermind behind setting up the system, and the rest of the lab, who's now still an amazing group to work with. And so I thank you for your attention, and I'm ready to take some questions.
Awesome. I'll give people time to raise their hands if they want to ask their questions out loud. And we've already got several in the Q&A box, so I'll start with a question from Emmanuel. To what extent do you think the promoter of the target gene influences the activity of the enhancers?
A lot. So yeah, so we have fixed the promoter as a minimum TK promoter. So this is just 70 base pairs to really make it as activatable as possible. We also think that it might be one of these promoters that could be activated by multiple different enhancers, and has been. But we think it's one of the parts in our system where we could really use some changes, and it's a direction that we're going right now to then also make the promoter exchangeable, and try to figure out how much promoter enhancer compatibility, and also how different promoters react to different enhancers. So there's a ton to understand.
So we have another question about whether or not the various enhancer combinations have differences in transcriptional bursting. And I guess I'd follow up on that to ask about how with your M-cherry fluorescence, it seemed like all the enhancers you look at basically have a unimodal distribution of activity. Yes. Whereas I know in other circumstances or with endogenous genes, you see a more bimodal or more complex distribution of transcriptional activity. Do you think something about the way you've engineered your system has resulted in a more uniform induction of transcription, or can you comment on that?
Yeah, so I think one big advantage is that we're always working on only one allele, right? And I think so yes. Yeah, so that I think is already taking a lot of the noise and a lot of the bimodal or potential bimodal distribution, or might take it away, because ours is really, then we're using mouse embryonic stem cells grown under 2I conditions. And so I cannot think about any other cell system that is as homogeneous as this system. And so I think that also adds to this homogeneity, or this what we're seeing. And then for the bursting part, that is actually the PhD project of Maria over here. So yeah, we're looking into that. So what we think, so we haven't looked into the combinations yet, but what we see for some of our enhancers, or for at least 2 different examples that we have, is that their mean expression is very similar, but the distribution seems to be different. And so we think this might be going back to bursting. And so we are now teaming up with Tineke Lenstra, and we're doing a lot of bursting analysis on these different cell lines. So yes, I think there is something, but I can't put in words what it is yet, because we don't have the data yet.
Sure.
Yes.
So then here's a question from Sunil. Is there how much or how little of the information is transmitted to the promoter through the DNA strands, like torsional or like twisting, sort of related to the first talk?
Yeah, so I actually don't think directly, because I think the distances are much wider. When we're talking about short distances, and I would say even within 200-300 base pairs, I would probably say this might be somewhat the case. But one of the reasons why we moved the first position at 1.5 kb is really to avoid these torsional stresses and not interfere with the promoter in that way. So I cannot completely rule it out, but I don't think this is directly translated to it here because the distances are just longer.
And so then I guess in a follow-up to your circumstance where you treated or where you deleted CTCF sites, Srinivasulu asks whether or not having stronger weak enhancers within an actual TAD or a boundary would have different effects as to whether they're in the same TAD or whether they're different TADs.
So I don't know. So if they are in the same TAD, so I don't know what would happen if you place them in different TADs. This you have to probably ask the CTCF and TAD experts. We're still at the very beginning of that part. I cannot answer you that. We don't think that we have actually different TADs here, but yeah, I cannot answer that. I don't know. We would have to test it.
Yeah. Have to find a whole new locus, I guess.
Well, we can just like we were thinking about putting in a TAD boundary or building a TAD boundary. So that would be something that we could do and see how that affects it. But yeah.
Do you think that have you looked at everything you're showing here is upstream of the TSS? Do you have anything to tell you whether or not upstream enhancers act differently than intronic enhancers?
So intronic enhancers. So we did work on intronic enhancers for the FGF5 project. And for that one, we moved the same enhancer from an intronic position to an from the intronic position to a position in front of the promoter at the same distance. And we didn't see any difference in how it affects transcription. And so we haven't done that yet. What I would love to do is to put the same enhancer upstream or downstream of the gene. So we would have. So we want to expand the system, put in a few more landing pads slightly outside, and see whether that would make any changes. But yes, it would be lovely to do that.
Awesome. Well, in the interest of time, I'll ask one more question here from Petros. And he says, for your distance-based model of enhancer activity, do you think that looping or a looping model or like a transcription factor diffusion model better fits the sort of relationship you're observing? Do you have any?
Well, I don't know. So what I think, so I'm not sure about any of the looping models. I would tend always to agree a little bit more with any kind of diffusion model. Of course, cohesin does play a role, and cohesin constantly wiggles around our genome. And so I really urge all of you to read Adrian Bird's recent perspective on molecular cell. I loved it. I thought it was really nice and really a fun read to just give a different idea of how it could be affecting gene expression. And so we know that cohesin for longer distances does something if there's individual enhancers. But if you, for example, look at the work from Elfesh Nora, where he just really beautifully, showed that at some loci you have these intermediate enhancers, which might be very similar to what we're seeing, that now give an enhancer the ability to activate even in the absence of cohesin. And so I think these ones might coming together. I wouldn't go with a condensate model, but there might be interactions that we might be seeing that might be only there and if we have numerous transcription factors and other factors bound there to then really lead to a higher local concentration and thereby lead to an activation of the promoter. So I think that would be a model I would prefer right now, but yeah, I can only hand wave right now.
Yeah. Well, thank you so much, for handling so many questions.
Thank you.
I'd like to again thank both of our speakers, Christa and Julia. Just a wonderful session, and I'd like to thank all of you for attending. And we will see you back here at the same place and same time in 2 weeks. So thank you, everyone, and have a wonderful rest of your day.